25 cycles, showing that the cells had been successfully removed. And also there was no traces of any MHC antigens, which would be the antigen responsible for rejection by the body. But very importantly, despite the fact that all of the cells had been removed from the trachea, it still maintained normal biomechanics. And this is very important for the long-term functioning and the immediate functioning upon implantation of the new tissue. So, while the donor trachea was being prepared and the donor cells were being removed, at the same time we had to prepare Claudia's own cells to repopulate this scaffold. So the trachea consists of an external framework of hyaline cartilage and then an internal covering of epithelial cells. So we needed both chondrocytes and epithelial cells to generate the new tissue. So this is where we became involved in the project due to our expertise in using stem cells to produce chondrocytes. We basically used a very similar method to the one that I showed at the first half of my talk that we developed for treating patients with osteoarthritis. So again, we took a bone marrow aspirate from Claudia's iliac crest, and then we isolated the mesenchymal stem cells simply by their adherence to plastic. We then expanded the number of stem cells that we had by growing them in the presence of basic fibroblast growth factor. And then at this point we carried out characterization of the stem cells to ensure that they were really behaving like mesenchymal stem cells, that they had multi-potential. You can see we could successfully turn them into cartilage, into bone and into fat in the lab under appropriate conditions. They also expressed characteristic cell surface markers of mesenchymal stem cells. So, having grown the stem cells and validated them, the final step was to promote chondrogenic differentiation. So this we did using our TGF-beta, insulin and dexamethasone system. And we also included parathyroid hormone-related peptide, as we'd shown in previous work that PTHrP can prevent hypertrophy of chondrocytes and prevent them turning into bone, which obviously we didn't want to happen in the new tissue. So while the chondrocytes were being produced, we also had to produce epithelial cells, and this was done also at the University of Bristol by a separate group. So to get the epithelial cells, the first step was to take a biopsy from Claudia's healthy right bronchus. Trypsin was used to disrupt that biopsy to release the epithelial cells, and again they were grown in the lab to expand the number of cells that we had available. And then they were characterized and to show that they express cytokeratins characteristic of epithelial cells, and also there was no sign of any fibroblasts in these cultures. So we developed Claudia's own chondrocytes and epithelial cells. The next step was to seed her cells onto the prepared donor trachea. And this was done in a specially designed bioreactor that was developed in Milan, in Italy. So this is the donor trachea, which had had all of the donor cells removed. That was then placed into this bioreactor, and this bioreactor was unique in that the two different cell types could be seeded onto different surfaces of the donor trachea. So Claudia's chondrocytes, they were seeded onto the external surface, whereas the epithelial cells, they were seeded onto the internal luminal surface. The other thing about this bioreactor was that medium specific for the two different cell types could be used in the two different chambers. So medium for the chondrocytes was placed in the outer chamber, and medium specific for the growth of the epithelial cells was placed in the internal chamber. The chamber was then sealed and left under static conditions for 24 hours. This was to allow all of the seeded cells to become attached to the donor trachea. And then after 24 hours, half of the medium, tissue culture medium from each of the two compartments was removed, and the tube was then rotated around its longitudinal axis. This was to allow oxygenation as it moved between the gaseous phase and the liquid phase. And the rotational force also provides the shear forces necessary for cell growth and differentiation. The tissue culture medium was changed periodically to remove waste products and to provide new nutrients. And it was left in here for 96 hours before the operation went ahead. So immediately before the operation, following the seeding procedure, scanning electron microscopy was performed to look at the seeding. So at the top we've got a picture of the unseeded trachea, and then at the bottom you can see that following the seeding you've now got a continuous coverage of cells over the surface. Also, cellular brushings were taken from the outer surface following the seeding procedure. And interestingly, you can see that the presence of chondrocytes shown in green, but you can also see the presence of a few epithelial cells. So if you remember, the chondrocytes were seeded onto the external surface and the epithelial cells on the inner surface. So it seems that you're getting migration between the two surfaces following the seeding procedure. So having seeded Claudia's own cells onto the donor trachea, the final step was the implantation procedure. So in June last year, the operation was carried out to remove the diseased bronchus and to replace it with a tissue-engineered trachea. And this was carried out in Barcelona in Spain. So this is a bronchoscopy at the time of implantation. And the thing to note is that the tissue is quite white in colour, and at the moment it clearly doesn't have its own blood supply. But the implant functioned immediately upon implantation, mainly due to the tremendous skill of the surgeon, and there was no signs of any air leakage right from the time of implantation. So 4 days after the operation, cellular brushings were taken again, but this time from the internal luminal part of the graft. And again you can see the presence of both types of cells, the chondrocytes and the epithelial cells, again showing this migration between the surfaces. This bronchoscopy, which was taken 2 weeks after the operation, you can now clearly see that the graft is quite red in colour, and it's obviously developed its own blood supply quite quickly following the implantation. And this is very important for the long-term health of the graft. Also, at 2 months after the operation, several biopsies were taken, and again you can see the presence of blood vessels. And this new angiogenesis, as I say, is very important. Okay, so the outcome for this patient was that the function of her left bronchus was restored, and her left lung was saved, and she did not have to have her left lung removed. And you can see from these two pictures how the diameter of the bronchus has been repaired on both sides here, and the airflow to the left lung was then restored. But importantly, there's been no signs of any immune rejection by Claudia's body. It's over a year now since the operation, and she's not shown any signs of any anti-donor antibodies, mainly because it was made from her own cells. So Claudia is now enjoying a normal life. She's been able to return to work, and she's able to care for her 2 children. So in this final video, we have a summary of the whole procedure. So this is Claudia before the operation. You can see her collapsed left lung, the damage to the left bronchus, and the severely reduced diameter, stopping the airflow to the left lung. So to make the new tissue-engineered airway, the first stage was to take a donor trachea from a deceased donor. And then all traces of that donor's cells had to be removed using detergents and enzymes. And that basically left a collagen scaffold. Then had to take Claudia's own cells, so we took bone marrow and turned them into chondrocytes, and stem cells into chondrocytes. And we also took epithelial cells. These were then expanded in the laboratory to increase the number of cells that we had. And then they were then seeded onto the donor trachea in the specially designed bioreactor. The chondrocytes seeded on the external surface and the epithelial cells on the inner surface. The final stage was then to cut the new tissue to the right size and shape, and in the end a segment of about 6 centimetres in length was actually implanted. So the final step was to cut away the diseased portion of the trachea, of the bronchus, and replace it with the